Good evening, everyone. Welcome to today's CellTalk webinar. Today, our speaker, Dr. Nupur Biswas, will be delivering a topic on investigating nutrient biomarkers of healthy brain aging, a multimodal brain imaging study. So, Dr. Nupur is working as a senior bioinformatics scientist at RENEX. She is working on bioinformatics and other multiple projects as well. Some of her projects include designing personalized neoantigen vaccines for cancer patients and exploring the genes behind chordoma and the health applications of digital healthcare variables. So Nupur has a doctoral degree in physics and her research works focused on studies of confined polymers. Before joining the RENEX, she worked at the Indian Institute of Science, Raman Research Institute, and Collab Tree, and the Indian Institute of Chemical Biology. At RENEX, she has gained experience as not only a research mentor, but as a scientist contributing to various international publications. Thank you. So I welcome Nupur ma'am to present this presentation. Thank you.
Thank you, Chair, for your nice introduction. Welcome everybody into the center presentation. Today I am going to present a paper which is authored by Zoelik et al. It is titled Investigating Nutrient Biomarkers of Healthy Brain Aging, a multimodal brain imaging study. This paper is related to the cognitive health in aging. It is published in this year in the month of May in the journal Aging. This journal has an impact factor of 5.0, and this research has impact on nutrition and cognitive neuroscience. We all are aging. Aging is a fundamental process of all living things. It is associated with gradual loss of physiological integrity along with the cognitive impairment. It induces impaired function of organs and with vulnerability to age. So understanding aging is a long quest for human beings to achieve the longevity, or more specifically, healthy longevity. And on the other hand, nutritional cognitive neuroscience also indicates that diet and nutrition promote healthy aging. And this paper is mostly concentrated on the cognitive aging. That's why it is related to brain aging more. And dietary markers, specifically Mediterranean diet, are associated with healthy brain aging. And now, as we age, our cognitive performances and brain functions decline. It is caused by the decline in cerebral volume, synaptic density, cortical thickness, and surface area and white matter fiber integrity. The concentration of metabolic markers in the neuronal integrity also declines with age. And this age-related changes also vary within the population. That is, it varies from person to person. And chronological age differs from the brain age. The trajectory of the chronological age differs from the trajectory of the brain aging. Now, coming to the nutritional background, nutritional interventions can cure aging process. Specifically, we are more interested in the Mediterranean diet. Traditional food culture that has developed around the Mediterranean basin is referred as the Mediterranean diet. And it is also noticed or studied that people living in these regions tend to be healthier and have a lower risk of many chronic diseases like cardiovascular diseases, compared to the people which follow other diets typically referred to the standard American diet. Now, Mediterranean diet has some features. Like there are people, as this diagram shows, this pyramid diagram shows, the people has in the daily consumption, the people having Mediterranean diets take lots of whole grains, breads, vegetables, fruits, and they consume a lot of extra virgin olive oil, which is actually the source of their fat. They take fish few times a week, or even less frequent consumption of the dietary products and more frequent consumption of the red meats, and with a very moderate consumption of the wines or alcohols. So this Mediterranean diet is an interesting fact to the researchers because of the people who living in that area have acquired some more healthier and lifestyle. With this background knowledge, the challenge is there are multiple trials which are investing on the role of the fatty acids on brain health, but the results are diverging. They demonstrate both positive and null findings. On the other hand, results related to the effect of antioxidants showed favorable effects on episodic memory, but not to the other types of memory which are affected by aging. Episodic memory refers to the memory through which person recollects some special events of his or her life. And there is another challenge which is related to understand the causality between the diet, nutrition, and brain health, and their role on the age-related neurological decline. So, the objective of the current research article is said to identify the distinct phenotypes of accelerated brain aging group and delayed brain aging within the older adults, and then compare performance measures of several cognitive performance like intelligence, execution function, memory in these two phenotypes, and then capture the distinct nutrient biomarker profiles within the observed phenotypes. So for this study, the researchers enrolled 100 healthy elderly adults from the Illinois brain aging study cohort. These participants were neurologically healthy without cognitive impairment, but they may have other comorbidities. Their age ranged within 65 to 75 years. Their brain MRI data was collected and processed. Their fasting plasma was collected to specifically assess three major classes of nutrients, which include fatty acids, carotenoids, and antioxidants, and vitamins. We first analyzed the MRI results, and we consider total 139 variables of brain health which are collected. These are enlisted here. These variables actually belong to three broad categories. That is brain structure, functional connectivity, and brain metabolism. Brain structure refers to the measured actually brain volumes from 68 regions of the brain. It includes both left and right hemispheres. Coming to the functional connectivity of the brain is actually mathematically modeled as a network. There are different networks are considered. These networks actually composed of the different functional regions of the brain, which these regions may contribute to a specific function. So these different types of networks are actually constructed in the brain. The researchers consider 8 such brain networks and they measure 4 separate block theory metrics, which are global efficiency, local efficiency, small volume efficiency, strength. And then they also measure the metabolites concentration in different regions of the brain. For each region, they measure 3 particular metabolites, choline, creatinine, and NAD. This metabolites, NAD actually, they will be regulated as with the neurodegeneration. So, through these variables, using these variables, they did hierarchical clustering and identify 2 distinct phenotypes. One group has optimal brain health and other group has more optimal brain health. So they named them as the delayed brain aging and accelerated brain aging. In the upcoming slides, we will talk about these 2 phenotypes, delayed brain aging and accelerated brain aging. Here are the average results of the different parameters they measured from the MRI images. So coming to the brain structure, it shows that the delayed aging phenotype have larger brain volumes with increased white matter integrity and increased concentration of brain metabolites, increased functional connectivity for the whole brain, and higher small wall propensity. Whereas the accelerated aging group has increased network functional connectivity for strength, local, and global efficiency. Then we compare brain age with the chronological age. they observed for the accelerated phenotype, average brain age is 65.1 years. This is brain age, and this like this chronological age. And the delayed for the delayed brain age group, average brain age is 59 years. So there is a gap of more than 5 years between these two phenotypes. And also we can see here clearly for the accelerated aging, brain ages are more closer to the chronological age. Here you see that the chronological ages are here, and it is listed to the brain ages, which is brain ages were quite low values. We further notice that there is a negative association between the brain age and different brain modalities. These modalities include brain volume, white matter drugs, brain metabolites, and functional connectivity measures. So, as these brain modalities are related to the brain aging, it can be used to the brain age and further refer to the delayed aging. Then the process measures and the cognitive performances of the person. They assess intelligence, execution function, and memory. So different parameters are referring to these three main cognitive behaviors are shown here. And this red plot is for the delayed brain aging with respect to the accelerated brain aging. That's why accelerated brain aging curve lies in the 0 values. And we see there are only two parameters for the delayed brain aging which are less values compared to the accelerated. And most of the parameters have this positive score. These positive scores indicate better cognitive performances for the delayed brain aging. Then further study the metabolite concentrations in the blood-based biomarkers. They collect, as we mentioned earlier, they collect in the plasma and then assess concentrations of the biomarkers. Nutrition status was examined based on that. So they identified 13 key nutrients which have higher concentration in the delayed brain aging phenotype. Right. This belongs to the fatty acid, the links are intestinal, antioxidant and carotenoids, and vitamins and some vitamin-like compounds. So this, as they identified these 13 nutrients, then they explored the literature, check their role in the aging process and our cognitive behavior. Among the fatty acids, acidic acid and butyric acid, these two are the monosaturated fatty acids. They are very common in the olive oil and Mediterranean diet. They are known for their antioxidant activities and they also enhance white matter brain integrity. Other food fatty acids, as shown here, alpha-linolenic acid, like EPA, EA, DA, they are known to reduce inflammation. And lignoceric acid, which is actually a long-chain saturated fatty acid and is found in the nuts or acid oils. It is also known for its role in the reducing of the cognitive decline. Among the other nutrients, carotenoids are known for photopigments. They are known for their antioxidant properties and benefits cognitive and brain health. Among the vitamins, vitamin E were identified and they promote cognitive and brain health, improves performance of working memory. Choline is not a vitamin, but like it's a vitamin-like component. It is a vitamin B-like nutrient. It is needed for the cellular structure and integrity of brain white matter tracts and volume. It promotes cognitive and brain health. So, what about the nutrients identified in this study are actually validated through the literature. So, the researchers observed the neuroimaging data shows the existence of two phenotypes. Delayed aging is inversely correlated with neuroimaging biomarkers of brain age. And the scores of cognitive assessments of intelligence, executive function, memory were higher for older adults with delayed brain aging. They discovered nutrient profile for healthy brain aging from nutrient biomarkers, not from dietary questionnaires. Usually, when the studies are held, but researchers ask questions about the diets of the participants or volunteers of the cohort. But in this case, the biomarkers were identified directly from the plasma. And they also identified the nutrient profile related to brain aging and clinically relevant neuroimaging biomarkers of brain age. Now, these studies have few limitations. Here, rule of long-chain saturated fatty acids and monosaturated fatty acids are in brain health are not understood. And cross-sectional, it is a cross-sectional study, sample size and the inclusion of the on only population participants is also those cells. It does not include any clinical trial which is needed to validate the roles of the nutrients. And more importantly, it identifies actually small number of nutrients, only 30 nutrients it identified. But and actually brain needs many nutrients for healthy functioning. So it may happen that it is missing some nutrients. Yes. So more studies are needed in this direction. In conclusion, we can tell that the study assesses brain aging through MRI data and cognitive assessments, identify nutrient biomarkers to promote healthy brain aging. It motivates future research and clinical trials. It shows the possibility of development of dietary strategies to support cognitive function and brain health in the aging population. The impact of the research lies in the fact that it identifies nutrient biomarker patterns that exhibit favorable relationship with cognitive performance and brain health. Thank you for your attention. If you have more questions, you can reach me at nupur@dennis.org.
Thank you, Dr. Nupur, for your detailed talk. Now, floor is open for the audience questions. So, here is the first question from the Vijay. Was the effect of genetic components and the comorbidities in brain aging evaluated?
Yeah, that's a good question. No, this study does not consider any genetic components or the genetic studies were not done. And regarding the comorbidities, the participants were free from the neurodegenerative diseases, but they may have other comorbidities.
Okay. Hope you got the answer, Vijay. I don't see further questions from the audience. Thank you for answering the questions, Dr. Nupur. Yeah. Before I conclude this webinar, I would like to inform you all that the next presentation will be given by Vijay Padul on trial of the mind diet for prevention of cognitive decline in older persons. So thank you for your kind attention and for joining the today's webinar. We appreciate your time and engagement. We hope you to see in the next webinar. Thank you.